Bismillahirrahmanirrahim. Assalamu alaikum student. Welcome back to your class of embryology. Today we are going to discuss with formation of the cardiac loop. So learning objective for the today's class are to understand the process of the cardiac looping and to know the embryologic origin of the different parts of adult heart. So this is the revision slide from the previous lecture, and you can see this is the area of the primary heart field, and primary heart field is forming the atria, the left ventricle and part of the right ventricle, while secondary heart field is forming the right ventricle and outflow region of the heart. So the heart tube continues to elongate as more and more cells are added from the secondary heart field, and this is very important for the lengthening process as well as process forming the right ventricle and outflow region of the heart. These two endocardial tubes then starts fusion in the midpoint in the center at the day 21, and when fusion is complete you can identify different area of this heart tube, and this is the truncus arteriosus, the bulbous cartis, primitive ventricle and primitive atrium. And then at the day 23 you can see the heart tube, the cardiac tube starts to bend in different directions. So formation of the cardiac loop, it is very important because it starts to elongate and fold on day 23. These are the different parts of the bulbous cartis. This is bulbous cartis, the conus cartis and the truncus arteriosus. This is the primitive ventricle and primitive atria, and these are the sinus venosus which are forming the inflow region of the heart. So when heart starts bending, and this bending will be completed by the day 28. So atrial portion initially a paired structure. So you can see these are the paired structure which are present initially outside the pericardial cavity, but because of the folding of the heart, these atria are incorporated into pericardial cavity, and these lines show the atrioventricular junction. These atrioventricular junctions remains narrow, and in the interior it is formed the atrioventricular canal. This area is the bulboventricular junction which later on form bulboventricular sulcus, and on inside you can see the interventricular groove. So bulbous cartis is narrow. You can see this bulbous cartis is narrow except at its this middle third proximal area which is forming the trabeculated part of the right ventricle. This is the conus cartis which is forming the outflow of the both the right and the left ventricle, and this is the truncus arteriosus, the more distal part which is forming the root and the proximal portion of the aorta and the pulmonary trunk. So junction between ventricle and bulbous cartis is bulboventricular junction, and it remains narrow. It is called the primary interventricular foramen. So as the cardiac tube begins to bend at day 23, the cranial part of the cardiac tubes move downward anteriorly and to the right side, while distal part or outflow regions moves upward dorsally and to the left part, in such a way that the common atrium which was present initially outside the pericardial cavity. So this dashed line area shows the pericardial cavity, and this common atrium is initially present outside the cavity. It is slowly now incorporated into the cavity, and it's come to lie behind the outflow regions. So embryological dilatations which are forming different structure in adult heart are the sinus venosus, which is the smooth part of the right, forming the smooth part of right atrium, the coronary sinus and oblique vein of the left atrium. The primitive atrium is forming trabeculated part of the right and left atria, and primitive ventricle forming trabeculated part of the left ventricle. The bulbous cartis is forming trabeculated part of right ventricle, its proximal part, while outflow part of the both the ventricle, distal part of the bulbous cartis is forming outflow of both the ventricles. The truncus arteriosus will form the aorta and the pulmonary trunk. So the different areas you can see that both heart tube have started to fuse in the center, and different areas. This is the outflow region, the future ventricle, the future atria, and after fusion you can see outflow region, the future ventricle and the future atria. Different factors are involved in the establishment of the heart field. So signals from the anterior cranial endoderm induces the formation of the induces the heart forming region in overlying splanchnic mesoderm, as a result of which BMP2 and BMP4 are secreted in the mesoderm. Another factor which is very important is WNT, and it must be inhibited, and the factor responsible for its inhibition are the crescent and the cerebrus factors, which inhibit the WNT. So inhibition of the WNT and secretion of BMP2 and 4, these are responsible for upregulation and expression of another gene, which is NKX2.5, and this NKX2.5 is the master gene for the development of the heart. Another factor, which is retinoic acid, also determine the craniocaudal axis of the heart primordia. This retinoic acid is initially secreted by the mesoderm in the splanchnic mesoderm, and this retinoic acid then induces a gene for the expression of retinal dehydrogenase. As a result of retinal dehydrogenase, the splanchnic mesoderm in the heart forming area in outflow region also start producing retinoic acid itself, and retinoic acid gradient increases in the venous area of the heart. As a result of increased concentration of the retinoic acid, this area is destined to form the inflow region of the heart, and lower concentrations are required to form the outflow region of the heart. So another diagram, you can say this is the fused heart tube, and in different parts, the outflow region which is attached to the first aortic arch, and the inflow part, then the myocardial mantle which will form later on the myocardial jelly, and then the pericardial cavity is present. This is the dorsal mesocardium, in which you can see the transverse pericardial sinus is present. Another diagram, before looping of the heart, you can see different parts of the heart tube: the truncus arteriosus, the common ventricle, bulbus cardis, common ventricle, and atria. And after the looping is complete at day 28, both the atria come to lie behind the outflow region of the truncus arteriosus in such a way that the truncus arteriosus now assumes a midline position, and both right and left atria bulges on each side of it. There are different clinical correlates which are related to the inhibition of the lengthening of the cardiac loop, as we know that more and more cells are added from the secondary heart field, and these cells are required for lengthening of the cardiac loop as well as forming of the outflow region. So inhibition of such process leads to the double outlet right ventricle. It can lead to ventricular septal defect. It can lead to tetralogy of the fellow. Can also leads to pulmonary atresia. So these anomalies are related to inhibition of the lengthening of the cardiac loop. Dextrocardia is another anomaly in which either it may be solitary defect or it may be associated with the situs inversus. In situs inversus, all the viscera are present in opposite direction along with the heart, which is present on right side of the chest cavity, and this is because of the reason that heart rotates in opposite direction. Dear student, this is a link for the animation of the cardiac looping, and you can best understand by opening up the link and going through the video. So the references are the Langman's Medical Embryology and Crash Anatomy. And now thanks for the present class. Until next class, Allah Hafiz.